All right, we're going to go ahead and we're going to finish out the macromolecules today. We only have 2 more to go, and so this should take less time than yesterday because we don't have all the intro stuff. So we have 2 left. We have our proteins and our nucleic acids. Now, both of these are going to be polymers, which means they're going to have a repeat monomer, and both of these are going to be critical for living cells. We can sum up the carbs and the lipids pretty easily, like carbs or sugars that give us energy, lipids or fats and oils that don't like water. But on this one particularly, it is really kind of hard to sum up proteins. Proteins have so many different types and so many different jobs. And so really the main thing we need to know about proteins, the for sure association, is that proteins are made up of amino acids. Okay, so this right here is the monomer for your proteins. And proteins are very specific when it comes to their shape. And all of these molecules really are. But something that we see over and over and over again is that structure determines function. And so since they vary greatly in their structure, they're going to vary greatly in their function. And so you can see that in terms of living cells or a body, they're everywhere. They're in everything, and they take part in almost every process that we have going on inside of our cells. So you kind of have just really not a complete list, but just kind of a basic list of some of the things that proteins can do. They can be structural, things like they make up our muscles, they're in our skin, they're in our hair. They could store some information, they could store, trying to think of some other proteins that might be storage. They're definitely transport, like hemoglobin is a protein that transports oxygen. They can transport things across the cell membrane. They're very important in cell signaling. In some future lessons, we're going to talk about cell communication, and proteins are critical when it comes to that. They help us move, like with muscles, with actin and myosin proteins that contract, and that's what allows us to move our muscles. Antibodies are proteins, so they help in defending us against pathogens. This is another big one, regulation, because that's going to be your enzymes. And so there's so many different types of proteins. We've got enzymes, which we're going to have a whole unit on those in metabolism. You've got things like keratin, collagen, elastin. All of those are some of those structural proteins. And hemoglobin, like I mentioned, that's one of your transport proteins. So lots of different types. Now, the one thing they all have in common is they are all polymers of amino acids. And so the basic structure of an amino acid is you have a carbon bonded to a hydrogen. Okay, over here you have an amino group. So there's one of your functional groups that you were supposed to know for chemistry. The amino group has nitrogen, and it has a basic quality, which is where you get amino acid. Part of it comes from that right there, the amino group. And then over here we have a carboxylic acid group, or a carboxyl group. And one of the things that this does is it gives it acidic properties, and that's where you get the name amino acid. Now, there are 20 different amino acids because there are 20 different R groups. Now, the R group is just kind of like a variable group. Different things can be found here, and depending on what you have in this location, it will give that amino acid different properties. So it could be some of these functional groups that we talked about. It could just be a single atom. It just depends on what the amino acid is. So here's your 20 different amino acids. And so the highlighted in the yellow here, or the green here, purple, or the blue, those represent your R groups. And notice they're each something different in that location, okay? And so the reason this is so important is because of the protein shape. And so it will always go back to the original amino acid chain, because the original order of amino acids is going to trigger different interactions among those R groups, which will ultimately lead to a new shape or a different shape in that protein. And this is why if we just change one amino acid, it could change the ending resulting shape of your protein, which if it's not, doesn't have the right shape, it's not going to work right. So it all goes back to these 20 different amino acids. Now there's 4 levels of structure in a protein. Okay, we call them primary, secondary, tertiary, and quaternary. And so this is definitely an area where I normally like to draw, but a primary structure is what's called a polypeptide. Okay, the primary structure is called the polypeptide. And so we have amino acids linked to amino acid, link to amino acid, link to amino acid. And normally a typical polypeptide is probably going to be at least 100 of these amino acids together. And so this right here is going to determine all of the other shapes that the protein takes on. So your polypeptide is just your basic protein. The protein actually doesn't work as a polypeptide before it becomes an active protein. It has to take on its final shape, but this is the precursor to that final shape of the protein. When we talk about protein synthesis, this is what we're making, a polypeptide. And the reason it's called a polypeptide is because the covalent bond here is called a peptide bond. All right, so this is going to be a dehydration synthesis reaction, and the bond formed between those amino acids is going to be between the amino group and carboxyl group. That's going to form what's called a peptide bond. Okay, so that's where you get the term polypeptide. There's many peptide bonds. And anytime you see pep, it's normally in reference to a protein. Okay, so for assay purposes, our monomer are going to be those amino acids. And here we have one of our bonds that we can find in proteins. That's a peptide bond. Now, what's going to happen next is that once we have this amino acid chain, depending on the R groups and the properties of those amino acids, they're going to start to interact. Some hydrogen bonds will form between some polar areas. They could have some other type of bonding that might occur. And what that's going to do is it's going to cause that chain to kind of twist like a helix in some areas, or it could also cause it to fold kind of like a fan in other areas. And so instead of just having a plain old chain, that chain is now going to twist and fold, and that's what we call our secondary structure, the twisting and folding. Once again, this is not going to be a functional protein. We're not done yet. So the next type is going to be the tertiary structure. And so once you have that twisting and folding, you'll have R groups that are now exposed to new R groups. And so you're going to have some more hydrogen bonding that could take place. You could have some ionic bonds that form within that protein. You could have some what's called hydrophobic interactions. And so when it comes to bonds on your proteins, you're going to want to mention more than one type. We have peptide, we have hydrogen, we have ionic. And so we could also have these sulfhydryl bridges, okay, these disulfide bonds. This goes back to that functional group called a sulfhydryl group. And so essentially what's going to happen is you're going to have all these interactions among those amino acids. And what you're going to get is what's called the tertiary structure or the globular structure. And literally it looks like a wad, okay? When you look up pictures of proteins, they're going to look like a glob. And so it just twists and folds and bonds even more to form the globular structure. Now, the quaternary structure is essentially the same as tertiary. A protein will only reach quaternary structure if there were multiple polypeptides to begin with. So like for instance, hemoglobin, it had 4 separate polypeptide subunits that make it up. And so that one would reach a quaternary structure. It is at these levels, the tertiary and the quaternary, where the protein is actually a functioning protein. So it starts as a chain, it twists and it folds, it globs up. If there's multiple chains, we'll hit the quaternary. But this is when the protein has its proper shape, and this is when it's going to function the way it's supposed to function. So these are a little bit better than my drawing. So you have the primary is the polypeptide, the chain. They twist and fold, they glob up, multiple polypeptide chains. Same thing over here, chain of amino acids, they can twist and fold, they glob up, functional proteins right here. All right, the last macromolecule are going to be your nucleic acids. Now your nucleic acids are the acids that we find in the nucleus, okay? But they are going to be a polymer of nucleotides. So this is going to be the monomer for our nucleic acids, and their main job is to store our genetic info. They hold the instructions for building our proteins. So what we need to make that amino acid chain is going to be found here in our nucleic acids, okay? They have the blueprint for making those proteins. And we have 2 main examples. We have DNA and RNA. Now these are pretty easy because the NA in both of those comes from this word right here. So this is deoxyribonucleic acid. This is ribonucleic acid. And so let's talk a little bit more about the nucleotides. Once again, the nucleotide is the monomer but there's actually 3 parts to a nucleotide. We have a sugar. It's actually a 5-carbon sugar, either ribose or deoxyribose. It has a pentagon shape, so that's the little house in your nucleotide. There's a phosphate group, which is normally just kind of represented with the circle. It's got a phosphorus and some oxygens there. It's one of your functional groups. And then it's going to have 1 of 4 nitrogen bases, or really 1 of 5, depending on whether it's DNA or RNA. And so your nitrogen base, it's a basic little compound that's attached, and your bases are the ones that hold the instruction. So we represent them with letters A, T, C, G, and U. You don't necessarily have to know this now, but A is for adenine, T is for thymine, C is cytosine, G is guanine, and U is uracil. Now, these nitrogen bases are either purines or pyrimidines. Okay, a purine has a 2-ring structure. So like down here in this nitrogen base, see there's a ring and there's a ring. So that would be a double ring structure. So that would be a purine. And then a pyrimidine just has 1. So there's going to be some pictures here in just a second. Now, hopefully from freshman bio, you remember your pairings. A always pairs with T, the apple's on the tree. And C always pairs with G, the car is in the garage. Now in RNA, we have U, and so U would also pair with A, okay? So one nucleotide has 3 parts. You have a 5-carbon sugar, you have a phosphate group, and you have a nitrogen base. And so there's a little bit simpler of a picture. You may want to do something like that for your activity that you're going to work on. But this right here is 1 monomer, okay? This monomer has 3 parts. All right, so the basic structure is your nucleotides are going to join up. The sugars and the phosphates join up to form the backbone. All right, and these are going to be held together by a phosphodiester bond, okay? On your essay, this is what it's looking for on the backbone, phosphodiester. And so that's going to be a strong covalent bond. But there's also going to be another very important bond in your DNA, and that's going to be a hydrogen bond. So remember that DNA is kind of like a twisted ladder. So the sides of the ladder are sugars and phosphates, sugars and phosphates. Sorry, it's kind of hard to draw, but. Now the inside are your nitrogen bases. You have A and T, and you have C and G. Now, this right here is where you're going to find your hydrogen bonds, okay? That's what's going to hold the 2 strands of DNA together. So we have strong phosphodiester linkages on the outside, and then we have some weak hydrogen bonds on the inside. And that's going to be important when we have to open this molecule up. And so we're just going to take a little bit closer of a look at DNA and RNA. There's 3 main differences between them. DNA has 2 strands, RNA just has 1. So DNA forms that double helix type of shape. RNA can take on different shapes. DNA has deoxyribose, which is where we get the D in the name, deoxyribonucleic acid. It's pretty much a 5-carbon sugar ribose minus an oxygen, okay? So they're very similar, just a very slight difference there. And then ribose is the sugar that you'll find in RNA, and that's where you get the R there, ribonucleic acid, deoxyribonucleic acid, that comes from their sugars. And then the other difference is in the nitrogen bases, where DNA will have thymines, RNA will have uracil. They're going to be similar as well, but slightly different chemically. And so in DNA, you won't find any U's. In RNA, you won't find any T's. And that is going to wrap up our macromolecule notes. So we have 4 main groups: carbs, lipids, proteins, nucleic acids. You're going to do some more practice, and you're going to have the rest of the day today as well as all day tomorrow to complete your macromolecule activities. I would encourage you to study for your quiz. Remember, it's going to be over both chemistry and water and macromolecules. But for this particular set of notes, all you have to do is I give you a statement, and you tell me which macromolecule group it's going to be in. So carb, lipid, protein, or nucleic acid. All right, I hope you guys have a wonderful day, and I will hopefully see you soon.